Hello, everyone. So here we are. It's finally time for ecology. Now, this is the first part, which is about relationships and energy flow. How cute relationships. So, let's get started. The first part then is just a few keywords and definitions that you need to know before we go any further in ecology. So, the first one is biodiversity. Now, biodiversity is a measure of all the plant and animal species found in an area. So, generally, if you have got a wide range of species in an area, we'd say that there is high biodiversity. Whereas if you have, for example, a field of just grass, because you've just got one type of plant, you're only going to have really one food source and one type of habitat. So therefore, you're probably going to have low biodiversity. So when you're thinking about biodiversity, it's important to think about what plants there is, because each plant can offer a food source and also different habitats to the animals that are found in that area. So for example, if you have a field of grass where there's only one food source and one habitat, you're only really likely to find a couple of things which can live in grass or feed on grass. So you're generally going to have low levels of biodiversity there. Whereas if you had something like a forest where there was lots of different trees in it, so for example, evergreen trees, deciduous trees, oak trees, sycamore, pines, all that sort of stuff, you've got lots of different types of plants. So therefore you've got lots of different food sources and lots of different habitats. So you're going to have lots of different animal species that can live there. So you'd have high biodiversity. Now, the next one that we've got is a population, which is a group of organisms of the same species living in one particular area. And a community is populations of many different species living together in a particular area. So, for example, if we take the forest as an example, within the forest, you could have a population of squirrels or populations of rabbits. But if you're considering those together within the forest, you could have communities or a community made up of squirrels and rabbits. The habitat then is simply the area which a community lives. So in this case, the habitat is the forest. The ecosystem then is an area where a community of organisms live and are affected by a range of environmental factors. So basically, it's how the organisms are interacting with the environment. Now, here's a little example then. So, this is a little pond. Now, in this case, the habitat is the pond. We've got several different organisms here. We've got Spirogyra, duckweed, which is a plant, pondweed. We've also got little fish, little tadpoles, and little water fleas. So, what we have got here is populations of all these different species. So we've got populations of fish, which is all the fish, so all the same species in that area. We've also got tadpoles. We've also got the pondweed. Now, they are a community because we've got populations of different species in the same area together. The ecosystem then is this entire pond where we have a community of organisms living and they're affected by the environmental factors around them. Now, the surroundings can also be described as the environment that they're in. It's the factors that act on the organism or the population, and we can divide these into two groups: biotic factors and abiotic factors. So the first one, biotic factors, are living things or living factors. So basically relationships between organisms within the environment. So we can have predator and prey, which is the example down here, where the bear is the predator, the prey is the fish. Or in this one, we have got the cheetah, which is the predator, and I'm gonna guess this is an ibex, but I couldn't be sure, and that's the prey. Competition for food would be something like bear versus bear, or leopard versus lion. There's also lots of other relationships like symbiosis, mutualism, parasitism. So there's quite a few. But the biggest one we talk about at GCSE is probably predator prey, and maybe even more than that, competition. Now, I'm showing you two pictures of animals here, but remember, plants can actually compete for different things, which we'll look at later in this. So even though plants might just look like they're sitting there, they can actually compete for different resources as well. Now, abiotic factors then are non-living conditions which can influence where plants or animals live. And we can have things like temperature, humidity, light intensity, and pH. So the next little part in this is all about sampling. Now, if you want to measure something about an environment, it's important that you understand what a sample is. So take this question, for example. Do these oak trees have the same length of leaves? Now, this one here is in a field by itself. This one over here is in a forest. So would they be expected to have the same length of leaves? Well, what you could do is you could take some of the leaves and measure them. Now, it's important to understand in this case that we would take a sample. Now, the reason we would take a sample in this is because what we could do is take all of the leaves and measure the length. But you'd probably realise, Alfie, what are you doing? But you would probably realise that it would take far too long. Okay, so we can't count all the leaves because it would take too long. So instead what we do is we take a sample. Now in this case, a good sample might be to take 30 leaves from each tree, measure their length, and work out an average. And that's pretty much how you take a sample. Now, within that, there's a few things we could talk about and we need to think about whenever we're taking samples. Firstly, a sample should be representative. So you should take as many measurements as possible in the time that you have. So what we've said there is, it's not possible to go out and count all the leaves. So you want to try and take as many as you can to make it representative. So, for example, maybe 30 to 50 leaves might be a good representative number to take for each of those trees. You also want to make sure that it is reliable. So what that means is that you should take lots of repeats and work out an average for the area. Then it also needs to be random. So you should use random coordinates. And that basically just helps avoid any bias. So it stops you just counting all the plants or all the features that you can see in one area. So when you're thinking about sampling, think of these three R's. It should be representative, reliable, and random. And this part just shows you how you can carry out random sampling. So if you imagine that this whole area is in a field, you basically take a measuring tape, start it at this point, run one that way, run another one perpendicular to it, and then use random coordinates. So you could literally just put the numbers 1 to 9 in a hat, draw them out, and in this example it's 5, 7. So they've literally pulled out the numbers 5, 7, walked 5 meters this way, and then 7 meters this way, and they count everything that is in this area. To do that, they use a quadrat. Now, one really useful piece of apparatus which is used in sampling is called a quadrat. There's nothing particularly fancy about it. It's basically a metal square, but the thing about it is that it's an area, or it's a square with an area of known size. So each side measures 0.5 meters, so therefore the total area of it is 0.25 meters squared. And later on, you can use that in the calculations of samples or working out how many of a certain species there is in a particular area, which we'll do a few examples of pretty shortly. Now this just shows us a little bit about how you sample plant populations. So sometimes what they'll use is percentage cover. So if you imagine that we've set up a field with our coordinates, we've walked out, and just keep going with the example. So we've walked out to the area, we've set our quadrat down, and these are the plants that we can see in it. What we can use is percentage cover. Now this is pretty much an estimate of what percentage of that square is covered by a particular plant. So in this quadrat, we're saying that roughly 60% of the area is covered by the large spiky plant, 10% is covered by the small spiky plant, and because we've got this one here, the 1% is usually given just to indicate the presence of a species. So it's not taking up very much space, but it just shows that it's there by giving it 1%. Now, I'm saying in that example that we use percentage cover. And the reason why we use percentage cover is sometimes it's not easy to count individual plants. i.e., it's difficult to tell where one plant stops and another one starts. So that's why sometimes we use percentage cover. But what we can also do is just count the number of plants. So pretty much, if it's easy to count them, you use that. Now that's sometimes called frequency or density. Now let's look at an example of this. So let's say that we go into a field and we count the number of buttercups. The field measures 30 meters by 20 meters. The quadrat had a side of 0.5 meters, 0.5 meters just, okay? Now, they take 3 samples, they count the number of buttercups. So in this case, it would be the frequency of buttercups because they're very easy to count. They take 3 samples and they work out an average. Now, let's have a look at how you would calculate this then. So what would the area of each quadrat be? Now the area. So it says each quadrat has a side of 0.5 meters. So 0.5, that's one side. And remember, it's a square. So 0.5 times 0.5 equals 0.25 meter squared. What is the area of the field? 30 meters by 20 meters equals 600 meters squared. Now, what would the average number of buttercups in a square meter be? Now, there's 2 ways you can do this. You can either realise that 0.25 meters squared on average contains 3 buttercups. So you multiply that by 4 to get 1 meter squared. Multiply that by 4 and that gives you 12 buttercups per meter squared. Or what you could do is divide the number of buttercups, so the average, by the area of the quadrat, which gives you 12 buttercups per meter squared. Either way, both ways gives you exactly the same answer. Now, the next part is estimate the total number of buttercups for this field. So what you've done here is worked out that there is 12 buttercups per meter squared. The field is 600 meters squared. So you have to do 600 times 12, which equals 7,200 buttercups. Now, just to link this with what we've talked about a little bit, this is a little sample. because they've gone out into this massive field and they've only counted a few areas. So let's think about the three R's. Was it representative, reliable, and random? Now, representative would mean that they could count as many samples as possible. Was it representative? Probably not, because there's only 3 samples, which probably means that it's not a very good indicator of that field. Was it reliable? So, not really. If it's not really representative, it's difficult for it to be reliable. Okay, they took a few samples and calculated an average, but there's just not enough samples. Was it random? Who knows? It doesn't tell you how they measured it. Okay, for it to have been random, they would have had to use coordinates. Now, another type of sampling then is belt transect sampling. Belt transect sampling is used in habitats where there is a gradual change from one side of the habitat to the other. The reason why we need to use it is because randomly placed quadrats would not identify the change in the habitat. For example, on a rocky seashore where the types of plants and animals change from the low tide to high tide areas. Now I'll show you a picture of that in a minute, but just to clarify here, we use random sampling when there is no clear zones and there is no changes in the habitat. So an example might be a field, whereas we would use belt transects for sampling if the zone changes in the habitat, or we can say there is zonation. Now in this, you basically set up a measuring tape here and move the quadrat to the first position. You count everything that's in here. So this can be used for plants and stationary animals. Then you move it down to the next area, count and record what's here, move it down, count and record what is here. Now let me show you some examples of some habitats which you could use these. So the first one here is a field, just this field here. Now would it be better to use random or a belt transect here? In this case, it would be better to use random sampling. So you would use quadrats and random numbers because there is no obvious change in the habitat. The next one here is at a water side. So here you have the water, here you have the moor, and here you have clay soil. Now what you can see here is that there's one type of habitat here, then another change in a habitat here, another habitat, and a fourth habitat. Down here again, you can see there's 1, 2, 3, 4 different types of habitats. So in this case, there's gradual change or zonation, so you would use a belt transect. This one again, this was the high and the low tide zones. So here is the high tide zone. So this basically means only when the tide is fully in will these organisms be covered with water. This is the middle tide zone. So that means they'll spend a good portion of their time underwater. And the lowest tide zone here, this part is basically where the tide pretty much covers these organisms all the time. So in this, there is clear zonation. So this one would have to be a belt transect. This large field then, even though there's these lines here, you can see that most of this area contains exactly the same organism. So there's no zonation, so therefore you would use random sampling. And this little fella just shows you how you could use a belt transect. So you set the tape up, you set the quadrat up at the first area, and then move along and count what's in it. And that's the end of this first little section. Thank you. Goodbye.